The treatment paradigms for non-small cell lung cancer are evolving. So starting in 2004, when EGFR mutants were discovered, a number of people tested for them, and some people gave the inhibitors of the epidermal growth factor receptor, preferentially those patients with mutants. We discovered in 2009 that the use of gefitinib, one of the epidermal growth factor receptor tyrosine kinase inhibitors, worked about twice as well as chemotherapy in the EGFR mutant population, and that it became a standard and approved both in the United States, Europe, and around the world to do the mutation for the epidermal growth factor receptor and to give those patients EGFR inhibitors. Then, starting in 2010, when we found out that patients with ALK rearranged tumors could more effectively be treated with crizotinib than with chemotherapy, that became a standard to test, and crizotinib then became first the favored therapy in second line, and then later in 2013, it was approved for first-line therapy of patients with ALK rearranged non-small cell lung cancer. Then in 2015, it was discovered that crizotinib was effective in ROS1 rearranged patients, and that became an approval. So with each several years, we've added additional testing, and we've added additional drugs. So for instance, now there's 3 different drugs approved for the initial therapy of EGFR mutant lung cancer: gefitinib, erlotinib, and afatinib. There are 3 drugs approved for ALK rearranged tumors: crizotinib, the first one, the second one is ceritinib, and the third one is alectinib. There's only a single drug now approved for ROS1 rearrangements, and that's crizotinib. When we treat patients with BRAF mutant non-small cell lung cancer, we think that we will now be evolving toward giving dabrafenib and trametinib, and it'll be the 4th genomic change that we see in non-small cell lung cancer, where there'll be a specific targeted therapy available for another subset of our patients. Dabrafenib and trametinib are currently available and are approved for the use in V600E mutant melanoma. The combination is currently being submitted to the Food and Drug Administration. We anticipate that it will be available in early sometime 2017 if the FDA rules favorably on the application for approval.
So the message for the community oncologist is that testing for BRAF mutation should be standard. It should be done at the time of diagnosis. You have the opportunity when diagnosing a V600E BRAF mutation of having a regimen that has approximately a 60% response rate. These responses can often be quite durable. The price of those responses in terms of toxicity is minimal in terms of severe toxicity. There are some management of grade 1 or 2 toxicities that needs to occur, but this can dramatically change the experience of a BRAF V600E mutant patient compared to standard chemotherapy. And the only way to identify these patients is to test. And if you test and you find, you must act with a targeted approach as shown in the phase 2 trial that documented the level of activity. The approval for this combination will likely be and should be in patients diagnosed with the BRAF V600E mutation, specifically for that subgroup of patients. This is an activating mutation. The trials were conducted in this population, and so that should be the indication. And given the level of efficacy that we've seen, coupled with a very attractive safety profile, I think this becomes the standard of care for all of these patients. And I think it then becomes standard to test for BRAF V600E because we have a therapy that can only be used if you diagnose this condition. The appropriate dosing for this combination is dabrafenib at 150 milligrams twice daily, and trametinib at 2 milligrams daily. There are appropriate dose reductions for toxicities if they occur, but that should be the starting dose and will be the FDA-approved dose for this regimen.